getting the links because
it's still early. Yeah.
he sent me an email asking for all the links and to be included in the mailing list.
I have him on my mailing list, so he should have received the
yeah, he has received it because
Okay.
he sent it to me. So
Right.
So
Great.
we're about to have elections next month, so it's probably best to do field work if we are already allowed after the elections.
Yeah, and it's also the research permit procedure has recently changed. I heard from Thomas.
Yeah. Yes, it has.
Is it a good thing?
It's a work in progress. The process is still a work in progress, I think.
Yeah.
So
it's the same in Indonesia also. So I'm not sure.
Has it changed? Has it changed there in Indonesia?
Yeah. So Ristek doesn't do it anymore. If you go to the website, they're still working on it. But all the research institutions have been put together in one single super research structure. So Arcanas is not. Yeah.
So it's all under Arcanas now.
No, Arcanas is now under this big research. BRIN it's called. I forgot what it stands for, but yeah. So it's not clear what. Yeah, the Geology Museum is not part of that, fortunately so far. But yeah, we'll see how it goes. Shall we start or?
Yeah, let's start.
Maybe we can begin with the opening remarks first.
Yeah.
All right.
Let me welcome everyone. So, good afternoon, respectively, good morning, everybody, and welcome to our archaeology webinar series of the Department of Sociology and Anthropology and Traces Asia, and to our concluding lecture for this semester on the topic Islands of the Small Giants, Pleistocene hominin arrival and its impact on insular megafaunas by Dr. Gerrit or Gerrit van den Bergh. The webinar is jointly hosted by Aido Balboa, Mylene Lising, Dr. Rixa Fuentes, and myself. This webinar series is supported by the School of Social Sciences, the Anthropological and Sociological initiatives of the Ateneo, and the Areté and its Eduardo Aboitiz Sandbox zone. Thank you all for joining us today. Now, today we have again an outstanding scientist and one of my favorite researchers in our webinar. So please let me welcome Dr. Gerrit van den Bergh. Dr. van den Bergh is a member of the Center for Archaeological Science, School of Earth, Atmospheric and Life Sciences at the University of Wollongong in Australia. Trained as a geologist and vertebrate paleontologist, Gerrit has led and been involved in a wide range of research projects on the interface of vertebrate paleontology, quaternary geology, and archaeology, primarily in Southeast Asia. He's one of the best-known researchers on the prehistory of Southeast Asia and beyond, a prolific author, and has an outstanding reputation as one of the leading scientists in quaternary research, with numerous publications in top journals. He began his career in 1988 at Utrecht University in the Netherlands with a Master of Science thesis entitled Sedimentology, Vertebrate Paleontology, Marine Geology, before he pursued a PhD at the same institution on the topic of the late Neogene elephantoid-bearing faunas of Indonesia and their paleo-zoogeographic implications, which he defended in 1997. Gerrit has held various postdoctoral research fellowships and positions at the Royal Netherlands Institute for Sea Research and the University of New England in Australia. In 2008, he became a research associate at the University of Wollongong and was appointed in 2011 as the prestigious Australian Research Council Future Fellow at the University of Wollongong, where he's working since. Gerrit is leading and researching in numerous excavations and field projects, most notably in Indonesia and the Philippines, where he's working together with Mylene, with Tomain Chico, Marian Reyes, Kat Manalo, who has joined us, and others on the famous rhinoceros site in Rizal, Kalinga. He's a member of the teams that discovered the so-called Hobbit, Homo floresiensis in Lianghua on Flores Island, and the earliest known hominin sites of Wallacea in the S.E.A basin area on the same island. It is a privilege and a great pleasure to have Dr. Gerrit van den Bergh in our webinar today. And I will now hand over the microphone to Mylene, who can tell us a bit more about our distinguished speaker and their collaborative research in Rizal, perhaps. Thank you.
Thank you, Alfred. Good afternoon, everyone. Again, it's a great privilege to be here and with Gerrit, who is part of our Kalinga expedition team. He also did his PhD under John DeVos, who was also is a member of our team in Kalinga. And as Alfred has pointed out, Gerrit is an outstanding scientist, and it's really been such a great privilege to be able to work alongside somebody like this. And one of the most memorable seasons that we had in Kalinga is when we were able to travel with Gerrit up the road to Tabuk City and hoping to find or at least find signs of the volcano that could have where the ashfall covering the site could have come from. It was so wonderful to watch a geologist at work. In February 2020, during a break in the Homo luzonensis conference, there was an impromptu visit of all the visiting international scientists to the Kalinga site. And we were just so fortunate to have Gerrit with us at that time to actually talk to us about the site and its formation and the material that we have found there. Otherwise, it would have been me saying, "This is the site, nice, no?" But yeah, it was wonderful. And that was the last time I actually saw Gerrit in person. And we're looking forward to having everyone back again in Kalinga, hopefully sooner than later, including Paul, who is here, and hopefully Kat Manalo eventually. So I turn you over now to Dr. Rick Sarfuentes to give us an abstract about Gerrit's talk.
Thanks, Mylene. So welcome, everyone. I'll just read the abstract. In 2003, a late Pleistocene hominin, Homo floresiensis, was discovered on the island of Flores, Indonesia. Since then, evidence has emerged that pre-modern hominins were present on other oceanic islands located between the Asian and Australian continents, and long before the late Pleistocene arrival of modern humans utilizing watercraft. In 2016, dated stone artifacts were reported that provided proxy evidence for the presence of an as yet unidentified hominin by at least 200,000 years ago on the Wallacean island of Sulawesi. And in 2018, stone artifacts dated at 700,000 years ago from the Kalinga site in the Cagayan Valley in Luzon were announced. The following year, there was the announcement of another insular hominin, the Homo luzonensis from Calao Cave, located about 30 kilometers northeast of the Kalinga site. These early and middle Pleistocene hominin sites have also yielded fossil vertebrate faunas, while temporary land bridges have been postulated in the past to account for the hominin arrivals to these islands. It is now generally accepted that overseas dispersal of both hominins and vertebrates would have taken place. As the low diversity but highly endemic fossil faunas indicate, Flores, Sulawesi, and Luzon have always been isolated ecosystems. Different species of diminutive proboscideans and giant rats were part of the Pleistocene faunas on these three islands, with the larger islands Luzon and Sulawesi harboring a greater diversity in known fossil vertebrate species than Flores. For this seminar, Dr. van den Bergh will elaborate on the available evidence around the impact of Pleistocene hominins on these insular vertebrate faunas. Again, we welcome for this afternoon's talk entitled Islands of the Small Giants, Pleistocene Hominin Arrival and Its Impact on Insular Megafauna in Southeast Asia, Dr. Gerrit van den Bergh. Dr. van den Bergh, the floor is now yours.
Yeah, okay, thanks for that introduction. Before I start, I should mention that my research has been primarily in Indonesia, as you already mentioned, but the results of this work are of course part of a joint collaborative effort. So I want to acknowledge my colleagues from the Geological Agency in Jakarta and in Bandung, the Geology Museum in Bandung, and the National Center for Archaeology in Jakarta. So much of what I will discuss today is based on this collaborative field and lab research. And although I've also participated in research in the Philippines, it was a really a joy to be part of. My focus today will be primarily on the Indonesian islands of Sulawesi and Flores. So let's have the first slide. Yeah. So Wallacea in the title. First, I should make clear what I mean with Wallacea, and for that I borrowed this map that was recently published by Ali and Heaney in 2021. So Alfred Russel Wallace initially placed his boundary, his zoogeographic boundary, running between, let's see if I can find, So running here between Bali and Lombok, Sulawesi, Kalimantan, and then turning eastwards south of the Philippines. And this was not known at the time as Wallace Line. That was Thomas Henry Huxley in 1968 who introduced this zoogeographic boundary as the Wallace Line, but he draws the line passing between Palawan here and Mindoro, so west of the Philippines. And Wallace actually never agreed on this position. So, and that was already a bit a forebode that absolute zoogeographic boundaries are difficult to pinpoint and define. So the correct way of referring to the Wallace Line is as indicated in this map. So this way, and this would be Huxley's line. However, Wallacea is nowadays usually used for this oceanic part, these islands surrounded by deep seas in between the continental landmasses of Southeast Asia and Sahul. Initially, the Philippines, except Palawan, were also included in that, and I think that's the way I use it, because all these islands in Wallacea, they have not been connected with the mainland during the periods of low sea level, during the glacial periods. And all the animals, the land, terrestrial animals that live on these islands nowadays, they must have arrived somehow by overseas dispersal. So let's have a look at what we know about when modern humans first arrived in Southeast Asia and crossed the Wallace Line. So the oldest anatomically modern humans that we know to date are from Morocco, dated at 300,000 years ago from the site Jebel Irhoud. But it's much later that modern humans appear in the Southeast Asian region. So at the moment, the oldest evidence is perhaps from Fuyan Cave in Southeast China, where there's a whole set of isolated teeth that have been attributed to Homo sapiens dated between 120 and 80,000 years ago. But this age has been contested, so there's no, they may be younger. Then there's also Homo sapiens teeth from a cave in West Sumatra, Lidah Ayer, and that was dated at minimally 63,000 years ago. But again, Here the problem is that the teeth were excavated by Eugène Dubois in the late 19th century. And we can only assume that these teeth were really from this fossiliferous Brexia that was dated more recently. So, and well, taking a sample from those teeth could provide an independent age for these teeth, but yeah, they're in Leiden Naturalis, and there have been no permission so far as I understand. So, but I think it's safe to assume that modern humans were certainly present in Southeast Asia by 50,000 years ago, based on the fact that they had reached Australia by 50,000 years ago, and perhaps even earlier, at 65,000 years ago. And that has been suggested for the site Majid Bebe in Arnhem Land. But again, this age of 65,000 is also not yet fully accepted by consensus. And then, of course, there's also from Sulawesi, the parietal cave art that has been dated. So various sites, the oldest one from Leang Tedong, if I'm correct. They are dated at minimally 45,500 years ago. And then in the Philippines and in Kalimantan, the oldest evidence for modern humans is Tabon Cave and Nia Cave. But there's now this mounting evidence that modern humans were not the first to reach oceanic islands of Wollongong. And the debate has shifted from whether it was possible at all to how they did it. So I tend personally to favor the hypothesis that the first arrivals of pre-modern hominins on Luzon, Flores, and Sulawesi were accidental, for example, by small groups being washed in the sea by tsunamis and rafting on flotsam to islands, as kind of random process. But there's also some researchers that maintain that they traveled deliberately to these islands from the mainland, using some kind of watercraft, maybe even Homo erectus, who knows. Whatever the case, we know that hominins had crossed the Wallace Line and arrived on Flores at least 1 million years ago. And this is proven by well-dated stone artifacts from the site Wollosega, and we will see a bit more on that later. Recent dating efforts placed the Liangbua fossils in the late Pleistocene, so around 100,000 to 60,000 years old. And then the much older Homo floresiensis-like fossils from the S.E.A basin on Flores, from the site called Matemenge. These are dated to be, were estimated to be 700,000 years old. And yeah, small mandible fragment here in blue from Matemenge is projected on top of the Liangbua holotype of Homo floresiensis, same scale, so it's actually a bit smaller. And then on Sulawesi, the oldest evidence are these Paleolithic stone tools from a site called Talipu, dated to at least 118,000 years, but probably much older than 200,000 years ago. And we don't know what the maker of these artifacts look like, but the old age strongly suggests that a pre-modern hominin is involved. And then, of course, as you are all aware, Luzon has recently joined the group of islands with its own tiny hominin species, Homo luzonensis. And the prolonged presence of hominins on Luzon is then further evidenced by the butchered rhino sites, Kalinga, associated with stone artifacts and dated at 700,000 years ago. Okay, so that's the evidence from the the islands, hominins, modern humans and pre-modern humans, hominins, I should say. And the big question, of course, is who were the ancestors for these diminutive hominins from Flores and Luzon? And there are mainly two competing hypotheses. So one is that, well, some researchers, who performed cladistic analysis on Homo floresiensis, they tend to favor an ancestry from a small-bodied continental ancestor such as Homo habilis, or maybe even Australopithecine ancestor. So the small brain volume and some skeletal characteristics such as the curved phalanges of Homo floresiensis and Homo luzonensis, and plesiomorphic primitive wrist bones of Homo floresiensis, they all tend to support this hypothesis. But the problem is a bit that to date, there are no such early hominin taxa recorded from the Asian region. So then a second hypothesis suggested Homo floresiensis is an insular dwarfed form of Homo erectus. So the morphology of the cranium and the dentition appears to support a closer association with Homo erectus than with these more archaic hominins. And besides, Homo erectus, of course, is known from multiple sites on the nearby island of Java, but also in eastern China. And they span at least the last 1.2 million years, which is considerably older than the oldest dated artifactual evidence from Flores and Luzon. So, and I tend to make no secret of it, I tend to favor this second hypothesis, and perhaps the archaic skeletal features of Homo floresiensis and also Homo luzonensis could that maybe be attributed to a kind of evolutionary reversal enforced by the insular conditions that may have facilitated a return, for example, to climbing to trees, maybe being safe for Komodo dragons, or maybe because of a simple ecosystem on Flores with limited species, no mammalian carnivores, maybe they didn't need so much brain power. That's just a possibility. On the other hand, I should admit that recently these artifacts from this the Chinese Lus Plateau that were reported by Zhu and Denel and colleagues in 2017 it was, I think. And they reported artifacts dated at 2.1 million year old, which is older than all the hominin remains from S.E.A. So maybe a precursor of Homo erectus, who knows? So there's still various possibilities. But let's have a look at the associated fauna. So all the Wallacean islands with known megafauna fossils have yielded at least one species of a pygmy elephant, or large mammals such as elephants. They tend to become smaller over time on islands, a process called insular dwarfing. And to the right, some of the insular dwarf proboscideans and their mainland ancestors are depicted on the same scale. So in the upper left here, we have the straight-tusked elephant from Eurasia and its descendants, Paleoloxodon nigrensis here, and Paleoloxodon, the smallest dwarf elephant that is known, Paleoloxodon falconeri from Sicily, derived from this huge mainland ancestor. Then in the middle, Mammutus colombi from the North American continent, and its descendants, Mammutus exilis from the Californian Channel Islands. And to the right, Stegodon zdansky and the island form from Japan, Stegodon aurorae. So while elephants and other large herbivores such as hippos, they tend to become smaller over time on islands. Small mammals, on the other hand, tend to become larger. And these size changes in body size, they are known as the island rule. But as you can see, the degree to which island dwarfing occurs varies considerably. And it is still being investigated why island dwarfing of large animals occurs at all, but it must be a strong evolutionary pressure because it has happened over and over again to various degrees. Now, the advantage of being a large animal is that you can travel large distances in search for food. Maybe when there's a seasonal climate, food is depleted at one site, then they can move long distances. Elephants, they do that in Africa, and also wildebeest and gnus. So they can travel long distances because of their large size, and they are comparatively safe against large mammalian predators. But there is a trade-off, and that is that it takes much longer to become reproductive and produce the next generation of elephants. And on an island, the advantages are kind of being lost because traveling a long distance, the same long distance as on the mainland, you would cross an island like Flores multiple times. So it doesn't make sense. You don't need to travel far distances because we go from coast to coast. And so a population of large elephants will have depleted available food resources on an island quite rapidly. And it is generally agreed upon that island dwarfing has to do with the more limited food resources available on islands, maybe combined with the absence of mammalian predators such as tigers or lions, because they can't maintain a viable population on a small island. So in a study that I published with Sandra van der Geer and other colleagues a couple of years ago, we compiled data on the body mass estimates of these insular proboscideans and the island surface areas. And we found that there was a significant correlation between the size of the island and body mass. So the smaller the island, the smaller the elephants were. Now, zooming in on one of these islands, Sulawesi, there's mainly two fossil locales known, and they are both in the southwestern peninsula of Sulawesi. This is a paleogeographic map that was published a couple of years ago by Nugra and Hall based on geological information. And you can see that the southwestern arm of Sulawesi is a separate island, and the two main fossil sites that we know so far are from this southwestern peninsula. So the oldest fossil vertebrate record from this southwestern peninsula goes back in time to around 2.5 million years. It's called the Wollongong fauna, after the main river that crosses the southwestern peninsula. And the vast majority of the fossil remains from here are represented by large vertebrates from multiple, the same fauna from multiple localities. And the most common mammal was a large pig with enormous upper tusks, Celebogurus. It's now extinct, but fossils have also been reported from Luzon, suggesting that there was some faunal exchange north-south or south-north between the Philippines and Sulawesi. There were also two dwarfed elephants in the Wollongong fauna and a giant tortoise, and they are all now extinct. And all their ancestors must have originated from mainland Asia, not in Australia. So this is the open site Talipu in the Wollongong Valley, where we conducted a couple of deep excavations on top and at the base of a small hill, which you can see here on the right. And on the surface, this hill was littered with silicified limestone, chert, Paleolithic stone tools, literally littered with it. So all over the place. So we decided to do some excavations there, instigated by the late Professor Mike Morwood. And we went down up to 12 meters deep. And that was basically to avoid surface disturbances and make sure that anything we would find was definitely occurring in situ and not disturbed by vertical movements in the topsoil. So as you can see on the left image, the upper 4 meters of the profile consisted of fluvial conglomerates and heavily overprinted by tropical soil. But they contain many stone artifacts, some aberrated by fluvial transport and others in a more fresh condition. And here you see this the profiles of the 2 excavations. So this is the higher one on the top of the hill, and then at the base we have overlap. This green layer is a tidally influenced deposit. So there were tidal currents active at that layer when that layer was deposited. And this layer is dated with optical dating methods to 156,000 years ago. And then the top of this sequence, this fluvial conglomerate, is between 100,000 and 140,000 years ago. So all these artifacts represented by these red triangles, and here some selection of those stone artifacts in situ from that level. They are up to 140,000 years old. Now this then we came into a sterile interval, and then at the base of the hill, the sequence continues down into floodplain deposits, fine-grained silt, clay, with a few artifacts and also a few fossils. I guess we were lucky that we decided to dig there. We might have missed it. It's not a very rich locality, but we got artifacts here and fossils. One fragment of a fossil stegodon tooth, also canines and teeth of Coelabochurus, this large pig, and some crocodile. And problem was that optical dating didn't work in this interval. It was too old, so the background radiation was just too high. So we tried uranium series dating on the fossils that were associated with these artifacts. And the disadvantage of uranium series method is that you can only provide minimum ages, and the minimum ages of the fossils, they vary between 200,000 and 600,000 years ago. So based on the combined results from the optical dating and the uranium dating, I think we can safely assume that these oldest artifacts from the lowest, deepest part of the profile are at least 200,000 years ago, and therefore most likely not made by Homo sapiens, because Homo sapiens, there's no evidence so far that Homo sapiens was in the area around that time. So, one thing I should say, we also found one piece of a molar of a bovid, which is most likely the ancestor of the iconic dwarf buffalo from Sulawesi, the Anoa. So, but it was in this fluvial conglomerate, so could have been reworked from older deposits, but that's the oldest record of Anoa's ancestor on Sulawesi. So here, based on all the current evidence, and I haven't shown everything, but I have constructed this fauna succession scheme for Sulawesi. Only extant vertebrate groups are depicted or included, for which fossils are known from the southwestern peninsula. For example, there are many endemic rodents on Sulawesi, rats, squirrels, but only for one rodent, Pauromys dominator, there is a Pleistocene fossil record. So I have left out all the other small mammals. And besides, yeah, we haven't really done a lot of sieving when we were excavating this in the 1990s. So yeah, we probably missed a lot of the small mammals at the time. If we go to the oldest Wollongong fauna, so this fauna is relatively well known from multiple fossil localities where we excavated, but numerical ages are well, the ages are very poorly constrained because it was very difficult to find suitable material for dating. So the early Pleistocene age of the Wollongong fauna is primarily based on paleomagnetic data combined with marine microfossils in the underlying strata. And we have a few U-series dates, but they are minimum ages, so that doesn't help much. But we think, yeah, they are early Pleistocene. Also, the two proboscideans from the Wollongong fauna, Stegodon and a primitive Stegoloxodon, which is an archaic member of the Proboscidea with 4 tusks. And that also suggests that we are in the early Pleistocene here. And closely related fossil proboscideans are also known from the early Pleistocene of Java. Furthermore, the Wollongong fauna contains this giant tortoise, and then also the most common fossil species is this Coelabochurus, this large pig. And as I mentioned, a different Coelabochurus fossil has been recorded from Luzon, from the Cagayan Valley recently. So from the numerous sites that we have excavated with Wollongong fauna, it's important to mention that none of these sites has provided proxy evidence for hominin presence in the form of stone artifacts. And we are pretty sure that the Wollongong fauna predates the arrival of hominins on Sulawesi. Then the next stage, the Tanrung fauna, middle Pleistocene in age, based again on uranium series dates, not very strongly dated evidence, but at least the fauna is completely different. So the 2 pygmy elephants from the lower part have been replaced by large newcomers, a large Stegodon and a big elephant, a Paleoloxodon. The tortoise seems to continue in the Tanrung fauna, and also Coelabochurus. Maybe there's one single teeth of possibly a Babirusa ancestor, which is the other iconic animal from Sulawesi, still extant on Sulawesi. The Babirusa with these upward curving tusks. And then slightly younger we come at the Talipo site that I just showed you. The fossil evidence is limited, but that is the site with the oldest artifactual evidence as proxy evidence for these hominins, dated at between 100,000 and probably over 200,000 years old. And then, yeah, there's some fluvial terraces along the Wollongong River with fossils of Anoa and stone tools. Again, we have some dates, uranium series dates, minimum dates, 82 to 90,000 years old. And then the late Pleistocene fauna from the several sites in the Maros karst area, where colleagues from Griffith University have been led by Adam Brum and Maxime Aubert. They have been dating the rock art there, but also done excavations, and we have there the oldest evidence for Homo sapiens in the form of these parietal cave paintings dated at 45,500 years ago. They also, the late Pleistocene cave deposits bear evidence of the modern, the extant endemic Sulawesi fauna, but interestingly also a large elephant is present in the late Pleistocene deposits associated with stone tools in the cave site Leang Burung too, but unfortunately that the deposits are quite disturbed. So, but it's probably around 40,000, 50,000 years old, maybe. So there's clearly a faunal turnover, but it's quite a staggered faunal sequence. There's a lot of data missing, hiatuses, so it's very difficult to pinpoint what exactly caused these fauna changes in Sulawesi. So therefore, I would like to take you to Flores. I'm going with the time. Okay. So where we have actually a much better dated faunal sequence and archaeological sequence. So basically two fossil locales, Liambua, well known on the western part of the island, cave, with a sequence spanning the last 200,000 years. And then the S.E.A basin in central Flores, small basin surrounded by volcanoes, and spanning the period between 1.4 million years and 500,000 years ago. So the S.E.A basin, several fossil sites and archaeological sites. It's a depression surrounded by active and inactive volcanoes, about 20 kilometers in diameter. And I'll show some of the numerous sites in detail from oldest to youngest. So this is the oldest site in the S.E.A basin, site called Tanitalo, where we have a single widespread fossil layer, which is covered by a pumice tuff, like a mass flow deposit associated with a volcanic eruption. And in this, covered by this volcanic mud flow, is a very rich layer with fossils of primarily Stegodon, a very small Stegodon, 1 meter tall at the shoulders. It's named Stegodon sondari, and also a lot of giant tortoise shell fragments and bone fragments. There's also Komodo dragon, crocodiles. So reptiles seems to have been the primary predators on Flores at this time. And yeah, this layer, we have excavated it at several places. In total, we excavated 216 square meters of this layer, and we have not found a single in situ stone artifact. So I think it's pretty safe to say that by 1.4 million years ago, hominins were not yet present on Flores. But absence of evidence is not evidence for absence, as we figured out when we wanted to publish that. So since then, we have done some more excavations in fluvial conglomerates in the same time interval, but even though there is suitable materials for making artifacts, stone tools, no in situ artifacts, only faunal remains. So then slightly younger is Tanitalo 2, which is 2 meters higher in the sequence. It's a different taphonomy. So most of the fossil Stegodon sondari here are juveniles, about 90%. But what is shown here is a single individual of adult Stegodon sondari. And then another thing is that the giant tortoise in this layer is absent. Komodo dragons and crocodiles still continue at this slightly younger site. Then there is quite a gap in the fossil record, but the oldest evidence for hominin presence is the site Wollongong, 1 million years old. So a lot of artifacts occur in a layer that is below a layer that is very well dated, with argon-argon dating 1 million years old. And below that, there's this fluvial sequence with a number of paleosols with in situ stone tools, so they are slightly over 1 million years old. No fossils, no fauna, and that's to do with taphonomy. It's a fluvial conglomerate, not very suitable for fossilization, although you could find some fragmentary fossils, bone fragments in some conglomerates, but not lucky here, which is unfortunate because we don't know if this Stegodon sondari and maybe the giant tortoise were still around at this time at 1 million years ago. Then Matamenge, and this has been the main focus of our research in the S.E.A basin. There's 2 fossil vertebrate-bearing levels at Matamenge, both developed in similar sedimentary facies, namely fluvial deposits alternating with fine-grained ashy mudflow deposits. And we are in a distal volcanic apron, the foot slopes of a volcanic fan. So here the lower level, which we dated at 780,000 years ago, where we have a medium to large-sized Stegodon, Stegodon forensis, and to the right here is a reconstruction based on an assemblage of multiple individuals. We don't have any articulated skeletons, but we had so many skeletal elements that we could actually build a skeleton. So it's not a single individual, but the height of at the shoulder is about 2.3 meters, so much bigger than that older Stegodon sondari. There's also Komodo dragon here, crocodiles, and abundant remains of a large rodent, rat, Hoiromis. And there is also a fossil avifauna, including ducks, swans, waterbirds, and a small eagle, a Hieratus species, analyzed by Hanneke Meijer, my colleague, who specializes on bird fossils. And the avifauna, interestingly, indicates that we have an open environment, but also there are wetlands around the site Matamenge. And we also know that it was a kind of savanna-like environment at the time that this layer was deposited, because of stable isotope studies of tooth enamel from the Stegodons. It was Mika Puspaningrum who analyzed those remains. And it shows that these Stegodons were all grass eaters. Also, lots of artifacts in this site, but because the material is all reworked by fluvial action, it is not really clear whether hominins who made these artifacts actually interacted with the megafauna. Then the upper level, estimated to be around 700,000 years old, has a similar fossil fauna as the lower level, albeit we have less Bergh material, or it's not yet fully analyzed. but it also contains stone artifacts, and most importantly, the upper level has yielded various hominin remains. So, oh, sorry. Again, this piece of mandible from a fluvial layer 2 here, this gray thin layer. It's a very thin lens actually that's filled in like a channel, and there's also a handful of from molars, teeth from a Homo floresiensis like hominin, a bit smaller actually than Homo floresiensis from Liangbua. And this is just to give you an impression of the size difference between Stegodon sondari from Tangitalo, 1.4 million years old, and then Stegodon forensis from Matamenge, 780,000 years ago, this new immigrant to Flores. Now we have done, over the past decade, we have done a lot of correlations, study of the geology, chemical fingerprinting of volcanic tuff layers. And what you see here in yellow, that is this ignimbrite that I showed in the image of Wollongong, which we have dated at 1 million year, and we can trace that back throughout the S.E.A basin, so very handy for lateral correlations. And then here you see the Tangitalo fauna way below this ignimbrite, and about 1.4 to 1.3 million years old. Then the lower Matamenge level here, with lots of artifacts, fauna, but no hominin remains. There's another site, Collaboratory, with similar fauna, slightly older than Matamenge. And then we have the upper Matamenge layer level with Homo floresiensis. And it's important to note that all the fossil-bearing levels, actually, they are associated with these ashy, clay-rich, ashy mud flows, indicated with pale yellow. And apparently they were very good in covering surfaces and preserving bones that were lying on the surface at the time. So we are now focusing on those kinds of facies when we are searching for new localities. At the top of the sequence is lacustrine, lake deposits. There's no vertebrate fossils, no artifacts, but it continues until about 500,000 years at the top. Then quick step to Liangbua, and I won't show a lot of that here, but just the recent redating of Liangbua. The holotype, Homo floresiensis here, just below this erosional contact. It is now dated between 100,000 and 60,000 years old. Then the last stegodon occurs at this level here, just below this gray layer, which is a volcanic ash dated at 49,000 years ago. So that's the last date of stegodon. Stegodon in Liangbua is, by the way, much smaller than the stegodon in the S.E.A basin, Stegodon forensis. It's probably continuous lineage, but over time it has become smaller in body size. It's about the same size as the Tangitalo stegodon. And then Homo sapiens, the oldest occurrence of Homo sapiens in Liangbua is just below here, this erosional contact, thought to be 46,000 years old. It's two isolated molars, and the presence of modern humans is also based on a change in raw materials from Liangbua. So all the artifacts from this channel fill and then the top of this undisturbed interval, they seem to have a preference for chert as a raw material, and the Liambua team has interpreted that as evidence that modern humans arrived around 46, between 50 and 46,000 years ago. And that's a bit in line with other places. So it's the similar faunal scheme. I'll go rapidly through it. So we have this fauna here with giant tortoise and Stegodon sondari. Then the oldest artifacts here at Wollongong, slightly over 1 million years old. Note that the ages here are not on a linear scale. So then the sandstone member of the Alabula Formation with many different sites, all with similar fauna and with artifacts, and at one side also with these Homo floresiensis-like fossils. The oldest fauna had an open forest or more forested environment, because we know that the Stegodons were mixed feeders. They ate both grasses and tree leaves, whereas the Stegodon florensis from the S.E.A basin, from Matamenga, was a pure grass eater. So it probably became much drier around the S.E.A basin at the time. So yeah, we have this final turnover here, but unfortunately a gap in the fossil record of about 400,000 years. And that's exactly the time when the first hominins seem to have arrived on Flores. So it's very difficult to draw conclusions if the extinction of the tortoise or Stegodon sondari are actually related to the arrival of Homo floresiensis ancestors. We don't know. The hiatus is too big. And of course, we know that there was some kind of environmental change. It became drier. We have a lot of volcanic activity on Flores. A major eruption may have wiped out some of the megafauna on Flores. So, like usual, we need more fossil sites. Okay, then we come to Liambua, where we still have basically the same fauna, but like I said, the Stegodon has shrunk a bit. We still have Komodo dragons. The rats have radiated into multiple species. There are 7 rat species of various sizes. As far as I know, no DNA has been extracted from those to see if they are closely related, like on Timor, for instance. All the rats, even though they are very different in adaptation and morphology, they all seem to be more closely related to each other than to mainland rats, so suggesting local radiation, and that would also be expected on Flores, maybe. The avifauna from Liambua and also the stable isotope analysis suggest a mixed forest and grassland environment during the Liambua phase. And that changes drastically when modern humans arrive, because then Stegodon disappears, Homo floresiensis disappears. The rats, the endemic rats, don't immediately disappear, but over the later Holocene, they gradually disappear, and then modern humans introduce all kinds of commensal animals that may compete with the endemic animals. And at the moment, there's only one endemic rat left, as far as we know, on mainland Flores. And then in the Neolithic also, people introduce cattle, but also wild pigs, porcupines, deer, monkeys, and they are now living in the wild. They are feral populations. But of course, it's tempting to associate the extinction of the older fauna here with the arrival of modern humans. And the gap is only maybe 3,000 years between the oldest occurrence of Stegodon and the youngest, the earliest arrival of modern humans. But Thomas Sutikna and his colleagues in 2018, they suggested that an abrupt and statistically significant increase in the use of chert for artifact production occurred around 46,000 years ago here. And they postulate that this change in raw material sourcing characterizes a pattern that indicates the arrival of Homo sapiens 46,000 years ago. But I must admit that I'm not fully convinced by that argument, and I'll show you why. So this is one of the levels in Liambua littered with juvenile Stegodon bones and stone artifacts. And this is the interval between 4 and 5.2 meter depth is characterized by high densities of Stegodon bone, very highly fragmented Stegodon bone, but also stone artifacts, and some Stegodon bones showing cut marks. And this layer was in 2016 redated to between 100,000 and 60,000 years old. Now, let's have a look at the age structure of the Stegodon death assemblage from this particular layer in Liambua, which is also associated with Homo floresiensis. So elephants, they have this horizontal tooth replacement, and during their life, there's a succession of 6 molars that pass horizontally through their through each jaw, and molars, they start to wear down anteriorly, and then the occlusal wear surface they increases as the molars progressively move forward. So if you can identify which molar you have and you see in what stage of wear that molar is, you can assign each individual elephant or Stegodon molar to a 1 out of 43 dental wear age classes, and that's what I've done for Liambua. Here are just some examples. This is a very juvenile mandible with the first 2 molars, and here is a very old individual at the other end, where the last molar is totally worn down. There's no lamellar ridges left. It's worn down to the root. Now these dental wear age classes, they can be grouped into age groups, which approximately represent equal lifespans of Stegodon. And these lifespans, then they are given in African elephant years, because we don't really know what the lifespan of a Stegodon or a dwarf Stegodon, for that matter, was. But the Liambua sample, they comprised of at least 48 individuals based on the dental elements. Now, if we look at the age structure of Liambua here, then practically 90% of all the fossils from Liambua are juveniles, whereas there's very few adult individuals. And that is usually a sign of predation. So there's this selective mortality of juvenile individuals, because predators, they tend to concentrate on the easiest to catch juveniles. And it's very similar to an age profile from the upper Tangaitalo level here, also 80% juveniles, and then a few adult individuals, including a single carcass. Also predation, but no evidence for hominins, no artifacts. So this could be predation by crocodiles or Komodo dragons maybe. And I should note that of all the juveniles, we only find practically only isolated teeth. So the postcranial skeletons have been very poorly preserved, maybe because they were eaten at this Tangetalo site. This pattern is from Liambua, and the upper Tangetalo level is very different from what we find at Matamenge and the lower Tangetalo level, which is associated with volcanic activity. Here you see an age profile that is much more gradual, so still about 30 to 35% of the individuals are juveniles, but then progressively older age groups, there's less individuals present. And that often tends to point to a mass death event. So the entire population died because of some catastrophic event. And because it's associated with volcanic mass flow deposits, the logical interpretation is that a volcanic event killed them off. But what intrigues me is that Liambua, we have this enormous high amount of juveniles, plus evidence for butchering. We haven't found any evidence for butchering or cut marks at Matamenge, and not even artifacts at Tangetalo. So I'm wondering if this floor at Liambua, so this level here, if that is not already modern humans who have hunted the Stegodon and maybe also Homo floresiensis to extinction around 60,000, 70 to 60,000 years ago. So that's just hypothetical, but I just throw it up. And yeah, not many of the Liambua team agree with me on that, but yeah, that's what I would like to see. So yeah, just some conclusions. The available evidence on islands with dated fauna records suggests no correlation between the first arrival of early hominins, Sulawesi, Flores, and I haven't talked about Luzon here, but there's even fewer dated fauna than on Sulawesi. And yeah, there's no correlation between the arrival of hominins and megafauna extinction. On Flores, the fauna succession and the first arrival date of hominins is best constrained by dated fossil assemblages compared to Sulawesi. So we know between 900,000 and 50,000 years ago, there is faunal stability, despite explosive volcanism, climatic fluctuations during this Pleistocene oscillations, and the presence of these tiny hominins. So the megafauna, the mice, only occurs after the arrival of modern humans, maybe as early as 60,000 years ago, but yeah, certainly 46,000 years ago. And that seems to suggest that modern humans are the culprit there. But pre-modern humans, no problem, they live in harmony with the megafauna, at least on Flores. So I wanted to leave it to that because I'm already over time, but I'm happy to answer any questions.
Thank you so much, Gerrit. Thank you for this amazing talk about those exciting and very important sites. We can now open the floor for questions, but perhaps before, I would like to welcome Thomas Inchico, our dear colleague, who is heading the excavations and the research team in Rizal, Kalinga, where the famous rhinoceros was found. So hello, Thomas, nice to have you here.
Hi, Thomas. I can't see you. Oh, there, yeah.
Okay, so maybe since Thomas is also joining us, if I can start with a question before we give the floor to the audience. Can you tell us in brief about the relationships of similarities and differences between Matamenge on Flores and Rizal Kalinga in northern Luzon?
Yeah, so, well, first of all, the age is very similar. So Kalinga, 700,000, Matamenge also, so the two levels, the youngest level with the hominins is also estimated to be 700,000. So that's one similarity. They both have stone artifacts. What we don't have on Flores is rhino. So, and I think the smaller fauna variability in Flores is largely has to do with, or maybe has to do with the smaller size of Flores in surface area. So there's this relationship between the size of an island and how many species it can maintain. And Luzon and Sulawesi are both much larger in surface area than Flores. So on Luzon, of course, you have also stegodons, you have elephant, you have actually more than one type of elephant. There's a large stegodon, there's a pygmy stegodon, or there's an elephant. Then you have rhinos. You have already in the middle Pleistocene, you have bovids, Celebogurus. So a much more diverse fauna in Luzon, which I think is the largest size, but it could also be that the main migration of fauna has been from north to south, so from China to via Taiwan to Luzon, then Mindanao, and then further south to Sulawesi, and further south to Flores. And the further south you get, the more depleted the fauna, the less number of species gets to get to the next step. So that's another explanation, but yeah. And of course, in Kalinga, the evidence for butchering on the rhino is very clear cut. And even though we have tried our best to find evidence for butchering on stegodon at Matamenge, so far we have not been successful. So we have a lot of bone surface marks, but it's very difficult to prove that they are cut marks, for instance, because the material is all aberrated by fluvial wear, so that obliterates any clear characteristics that you would expect with cut marks on bones. So, and yeah, fresh bone breakage, shattering is also a bit problematic because almost all the bones are broken, some with fresh breaks, some with old breaks, but as studies from Africa suggest, recent elephants, they trample bones, especially when they are a bit covered with sediment. They don't see them, they step on it and it breaks. And I think we have had similar processes at Matamenge. And yeah, the site in Kalinga, there it's the material is much fresher. The state of the fossil material is much fresher. So the evidence for butchering is clear there. And we haven't been able to prove that for Matamenge so far. Does that answer your question?
In a very wonderful way. Thank you. Yeah, it's actually, it's indeed the Kalinga rhinoceros. That's really a wonderful find, and the appearance of cut marks and butchering traces over such a long period of time, and despite the many possibilities for post-depositional surface modifications, is quite awesome. Also that an entire assemblage of stone tools was found in the same context. And from the user analysis, it seems that some of the stone tools at least may have been involved in the butchering activities, but there were also other activities that could suggest that it's not just, how to say it, a kill site, but they stayed there for maybe a considerable time and doing other things aside from getting the meat of the rhino.
Yeah, maybe Thomas can elaborate more on that. If
maybe he can write in the chat box, because he just texted me that he's unfortunately has neither microphone nor video. So let's see if he's able to write something. In the meantime, I think we have other questions from the audience. Ado, am I right?
We do have one from in Zoom.
Yeah.
All right.
Okay. Yeah. Right,
So this is from Shen. Sorry if I'm mispronouncing your name. He or she says, thanks for the excellent talk. I have two questions about Paleoloxodon from Sulawesi. Is there any indication of dwarfism? What species of Paleoloxodon may that be?
Yeah, that's very. We have a few milk molar fragments, so it's very difficult. I think they are Paleoloxodon, but I'm not 100% sure. I must admit, it might be Elephas, but it's based on the double layered enamel. So Paleoloxodons tends to have a bit thicker. Yeah, it's a bit a technical thing, but you would like to have a complete skull, because Paleoloxodon has this very characteristic skull, but skulls are very rare, elephant skulls, because they are pneumatic bone and they get compressed, they are damaged very easily. So we haven't been able to find that yet in Sulawesi. But yeah, it's a large-sized Paleoloxodon. It hasn't become dwarfed a lot. So it could be a recent arrival. In the Tanrung fauna, we have some complete molars. They are definitely more primitive than Elephas maximus, the modern elephant. But again, no skull material. And yeah, relatively large size. But I don't know if the Paleoloxodon or the elephant from the Maros Karst, the late Pleistocene one, is actually the same descendant from the Tanrung elephant. So, because elephants, they are practically everywhere. They swim to islands and they live in herds, so they are excellent island colonizers.
Thank you very much.
Thank you. Meanwhile, may I read the reply from Thomas? So Dr. Inchiko says that Kalinga is primarily a butchery site, but might indeed be more than that. And he points to the example that there are several cores, lithic cores on the site, which you would not expect at first hand on a kill site. But then again, potentially these cores were brought in provision for a future need of flakes in the butchering process. And if so, then we would have a nice example in Asia of predetermination by early humans, which, and then he adds to that, which matches with the hand axe manufacture as evidenced in Arubo in central Luzon. Thank you, Thomas. It's actually great points. So the presence of several cores with the rhinoceros, it's quite intriguing indeed.
see
You
Dr.
see-
Lee was also present, but he has already gone.
Yeah. He writes that nice to see you all. Thanks for the webinar, but he has to go to attend another class.
Oh, okay. Yeah. Well, say hello if you meet him.
Paul.
Yes, hello. The Tangitalo 2 site, I'm intrigued by the absence of the large tortoises. Wouldn't that be a sign of possible arrival of hominins? Wouldn't the large tortoises be the first to go?
Yeah, that could be, but then again, we have no artifact evidence from Tangitalo, and we really have searched very thoroughly, also on the surface, and they are just not there.
But
So,
you also have no tortoises anymore, so
yeah,
if
well,
the tortoises were the first killed, then and you don't see the tortoises, then maybe the artifacts are also sparse still. Maybe you don't need that much artifacts to kill a tortoise.
no. Yeah,
Maybe you just can eat the eggs.
when I say artifacts, I basically speak as proxy evidence for hominin presence. And if there's no artifacts at all, then I assume that they were not present. Another thing is that we do have evidence that Komodo dragons ate giant tortoise. So we have one tortoise carapace and a plastron. And inside we find isolated Komodo dragon teeth. So it's a possibility that they, gradually wipe the tortoises out or to extinction. But yeah, I would really like to have more evidence for that.
You couldn't wipe tortoises out by eating the eggs.
Yeah, he did that, and of course, monitor lizards, they like eggs very much. So that's, it could have been that they were just preying on the sites where they lay their eggs, or yeah, that's possible.
But hominins could easily find or excavate eggs that are buried, or and so the tortoises would be possibly the first to go. So disappearance of tortoises might indicate presence of hominins.
Not necessarily by humans, but it could have been Komodo dragons. Yeah.
Thank you.
Thank you, Paul. Mylene, I think I interrupted you.
If there is no other question, I would like to ask what might be a very elementary question. So you were showing earlier, you were talking about the stratigraphy. What happens when in the stratigraphy you have layers with fossils and artifacts, and then all of a sudden there's a sterile layer, and then another layer that has, again, material in it? What happens when there's a sterile layer?
Well, as an example, that Talipu site in Sulawesi, you need a bit of luck. So you can imagine making such a deep excavation is a lot of effort. And we were just lucky that we had four flakes in situ in that deepest level. If we would have missed those, then we would have 3 species of animals from the lowest level and 1 from the upper level. But yeah, I guess we were lucky. But sometimes you need a bit of luck. On Flores, however, it is practically every site with fossils also has artifacts. But all those sites, except some of the paleosols at Wollongong, there are artifacts in situ, but no fauna, because bones on the surface of a soil, they don't fossilize if they are not covered with something. So, but artifacts, of course, they preserve. They're not like bones. So if hominins would have been present at some level, and there's suitable material for making stone tools, then my take on it is then you would probably find artifacts if humans would have been around. But it's very difficult. So I'm currently working on a program, so we need much more dates and then try to deal with that statistically. So, but yeah, it's difficult. Absence of evidence is no evidence for absence.
Thanks. So when a layer is sterile, it could be possible that it just so happens that you were not lucky enough to come across a bone or a fossil or artifacts, or
yeah, no, but
there's actually
let me,
something that's happening or that happened that, you know, there's a pause in all these faunas being present, or even hominins.
Now, basically, what we usually do is we search for new fossil sites by looking at bone fragments on the surface. And when we find it, we do a test excavation. And if there's more bone in situ, then we maybe do a larger excavation. We have done that now maybe at 10 to 15 different sites. And every time when there's bone and we excavate, then we find at least a few flakes or artifacts there. So they are very, actually in the S.E.A basin, it must have been a very prolific place to be, because there's so many artifacts in that interval from 1 million year to 700,000 years. There was water nearby, we know that from the ducks, and there was probably a big lake in the caldera north of the basin. There were permanent streams based on sedimentological evidence and mollusks. So it was probably a very good place for them to hang around. And yeah, the artifacts are everywhere, basically. Where we have fossils, we have artifacts. But proving that they were butchering the fossils, that's another story, because most of these sites, they are fluvial sites, and yeah, stuff is reworked. We have so far not found any articulated skeletal remains of Stegodon except for 3 vertebrae. But yeah, does that answer your question? So that's why I think that in the lower levels where we have fossils and we have suitable, but a lot of the artifacts are made from basalt, not very high quality material, some from chert, but mostly volcanic rocks. And they are present at Tangi Talo also in conglomerates, in fluvial channels, and no artifacts. So I think they were not there yet, but yeah, try to prove it.
Thank you. Yeah, that's true. That makes sense. Thank you. Alfred, there's a question from Thomas.
Yes, thanks, Mylene. Thomas is asking, Gerrit, what prevented you to name the S.E.A basin hominin Homo floresiensis?
Oh, well, that is because a lot of the diagnostic criteria, and I must say that was mostly Yusuke Kaifu's work. That's his specialty. But most of the diagnostic criteria for Homo floresiensis, the type specimen, they're actually based on skeletal elements. And we only have a few isolated teeth. And actually the first lower molar of the Matamenge hominin has still 5 cusps. It has a hypoconulate, and that is lacking in Homo floresiensis from Liangbua. So Liangbua has more derived M1. That's the only thing. I'm pretty sure that it's Homo floresiensis ancestor, but yeah. Ideally, you would like to have some postcranials to confirm. And we didn't expect such a tiny hominin at Matamenge, we were expecting a large-bodied Homo erectus, but yeah, that's probably a bit earlier in the sequence. So we were trying to find sites that are around 1 million year old.
Thank you very much. I guess that answers Thomas' question. So yeah, I would have also thought it would be a regular sized Homo erectus kind of hominin.
But we find actually something smaller than Homo floresiensis. But that's not the reason why we didn't name it Homo floresiensis, but just to be safe, Homo floresiensis like. I mean, you have to be very careful with hominins because people take offense very easily.
Yeah.
Paleoanthropology is a wasp hole.
I heard.
A wasp nest, yeah.
Wasp nest, yeah.
It's a Dutch expression.
All right. Do we have other questions in the chat box or on Facebook?
None anymore, Doctor. So,
And I think it's beer o'clock, at least here in Australia.
Oh, this is nice. Not yet here. I'm afraid I have to still attend another meeting.
Hang on, Mylene, I will call Susan. Susan?
Oh, because you are 2 hours ahead. They are 2 hours ahead of
Yeah,
us.
yeah.
So
It's almost 6 o'clock now.
Mylene wants to say hello.
So yeah, so Susan was with us also in one season, and I remember when she held this facial reconstruction workshop.
Hang on a sec, she is coming.
I was just saying that I remember the facial reconstruction workshop that we had under Susan one time here. That was so interesting. We all had the same skull, but we came up with different individuals, different looking,
Yeah. Different outcomes. Yeah.
different outcomes. Yeah. So you're 2 hours ahead of us, Gerrit.
3 hours, I think. It's
Oh.
yeah. What time is it there? It's here. It's 5:45 now.
Is it Mylene from Cagayan?
Yeah.
Ah, okay. Yes.
Hi. Yeah.
Susan.
There.
Hi, hello.
Hey. I can't hear you because he's got the thing inside.
Oh, yeah. I'll take this off. Yeah. Sorry.
Hey, Mylene, how are you?
How are you? Nice to see you, Susan.
Nice. Oh, you look so warm.
You look good. You're looking good yourself. Oh, it's cold there right now.
Yeah.
So
2 years of cold weather.
Rain.
Woo.
rain we have here too. Oh, we look forward to having both of you
Bad
back.
storms. Very sorry about that. Yeah.
Actually, right now, I turn my noise cancelling off. I don't know if you can hear it.
Already, Alfred.
Oh,
Yeah.
yeah. Okay.
Typhoon.
Yay.
It's actually already dark. Oh no.
Yeah.
Susan, we have to have your workshop again sometime soon. That was very interesting. The reconstruction, the facial reconstruction workshop that we had before.
Yeah, okay. I would
Yes,
love to.
yes.
Yeah, absolutely. It's been a long time.
Yes.
I've run those, yeah. Mostly now I teach statistics. Yeah.
Oh wow.
I
I'm
think-
scared.
But we
It was
both,
very interesting.
we kind of, think a lot of times about the times in
Cagayan and
Cagayan.
Manila, in Diliman, and
very
the
good
conference.
memories.
Yeah. Which of course we were there.
Yeah, was that like one of the last times that you were able to travel? Because that was
the last time we were able to travel.
That was the last time too I was in Cagayan.
Yeah. Also, yeah. Because I don't know if you remember, I got a cold. And then I went to the hospital, yeah, in
Yeah. In
to-
Tuguegarao, right?
that's the one. Yeah. But it was fine. But then, of course, when we flew back to Australia after Bandung, everything closed.
Right.
And for a little while there, we could only go 10 kilometers from where we live. So we know every bushwalk, every park. Yeah.
That's
So
nice though, that you were still
was-
able to go outdoors.
you could go for food, that was it, and for exercise. But it would have been the same in Manila, yeah, a little bit.
No, for us, for a while, we actually could not, we were not allowed to go out.
Yeah.
Yeah.
The base
We
home.
had a lockdown on March 16 of 2020. For
Okay.
a while, right? Rick, we were like, we could
Yes.
not go out. Yeah.
Yeah. We were only allowed one person could go shopping once a day
Same.
or once a week or something. So we learned. Yeah, it was very scary times. But the strange thing is that we now in Australia have got a higher infection rate than we've ever had.
Right now.
Yeah. So in